Hello, everyone. Good afternoon and welcome to the seminar Speaking with Scientists about Sensitive Research Topics, Animal Experimentation and Genome Editing. It's really a pleasure to have Pedro Hermes and Olsson with us. Good afternoon and welcome to the seminar. I would like to greet our speakers and those who support this initiative, in particular the research team of the project DataGov. The presence of those who are with us in the YouTube channel is really appreciated. I would like to ask you to write your comments and questions in the YouTube, and our speakers will answer them in the end of both presentations. In this seminar, we will discuss the ethical, legal, and social issues surrounding contemporary biomedical research based on the perspectives of senior researchers. Pedro Ramos will talk about the ethical implications of genome editing, the focus of his PhD project. Afterwards, Anna Olsson will reflect on regulation of animal research and the role played by animal experimentation in a One Health context. I will start with the presentation of Pedro Ramos. He completed a Bachelor of Science in Biochemistry at Faculdade de Ciências e Tecnologia and a Master in Molecular Genetics and Biomedicine in a partnership with the Institute of Molecular Medicine, University of Southern Denmark. He is actually a PhD student at University of Porto, conducting his research activities at the Laboratory Animal Science Group, coordinated by Anna Olsson in the I3S, Instituto de Investigação e Inovação em Saúde. Pedro, thank you very much for being with us today, and you have the word.
Okay, thank you, Susana, first of all, and thank you for the invitation to speak on this webinar. I think it's time for me to share my screen with all of you. And currently, you should be seeing a possibly blank space. The blank space is just a mere introduction to what I'm going to speak about, in the sense that what I'm bringing is not just some space for giving you results of our own work, but also a space for reflection. So I'd like you to think in this sense, to have a discussion, a healthy discussion with all the topics that we are going to bring here for you, as Susana already mentioned. In terms of the project that I'm working, and one of the two that are going to be presented today, it's a project where I'm inserted as a PhD. The name of the study that I'm talking about today is about ethical and technical challenges in genome editing, and it was a qualitative study conducted with researchers, biomedical and not just biomedical researchers, but also senior researchers that are working with animals, for example. And this study itself was conducted by me, and Anna Olsson, of course, is the supervisor of the project, together with Marie Strest from ICBAS, which is my co-supervisor, along with Cord Brakebusch, that is based in the University of Copenhagen. And the three of us, me, Anna, and Cord, are actually part of this network, this consortium named IMGene, which stands for Improving Genome Editing Efficiency. And as I said, I am one of the fellows that is part of such consortia, with Anna being my supervisor. And then I also have 7 other research fellow colleagues, which are working on a wet lab-based scenario, a little bit different from me, which is conducting a different type of work. And this is about, that is exactly about what I'm going to talk today. In terms of the genome editing topic, and I think this is a good idea to bring to light what is genome editing and what is CRISPR-Cas9, because I think it's a technology that has its emergence in the 2012, and it's actually one of the technology worldwide used nowadays. Most of the labs changed also their scope of research to using CRISPR-Cas9, most of them that used other studies of silencing, RNA silencing, for example, they changed to the CRISPR-Cas9 technology. So what is this technology has in particular compared with others? First of all, it's just a technology that allows you to do something that was not as easy to be done since the first 2000s decade. And what it allows you is that it may leave you to induce a cut on the DNA on a site of your choice with the aim to edit it. So it's something very precise. It's not random, as previous genome modification techniques were. And in that sense, it uses 2 different molecules that we can see here. One is the guide RNA, which has a sequence of nucleotides that will be complementary to the DNA of the cells that you'd like to change, to modify. And then you also have, this works like a blind dog or like a dog for blind people. So it will conduct the Cas9, which is a protein that has a scissoring activity. And the way that Cas9 works is when it identifies the cut site that we would like to change in that position, it will cut, it will induce a double strand break on the DNA, and then the cell will repair it. And because this cannot stay like this with an empty space. So then it will happen through 2 ways. One is non-homologous enjoining, where nucleotides will be inserted or deleted to fulfill the blank space. And then the other mechanism is HDR. So it's homology-directed repair. We provide an external DNA template, and we replace the blank space by this template that we'd like to introduce. So it works similar to what we have on our when we are writing in our text editors, that we replace one of the words by another one. So we replace, for example, here automatic text editor by automatic DNA editor. This works the same way, but with biological sequence. One of the 3 of the features that this Cas9 has is that, as I said already, has a lot of the same characteristics as previous techniques in terms of efficiency. Is also very straightforward, is very precise, and it's very cheap, on the dozens of dollars. And then one thing that is actually striking is that any organism is nowadays editable. So we don't need to continue working with the same animals or other species that we were working before. We can work with new ones, or we can work with others that we never thought about. But as I said, my 7 other colleagues are working to do this, to improve this pathway efficiency specifically. The reason is because we can control it better. We don't rely on something so random as the NHEJ pathway. So they try to balance on this pathway mechanism. Whereas I'm actually concerned what this will mean. Because a lot of unknowns might be brought here to light in terms of the application of this technology in different applications, both in humans when we talk about therapeutics and enhancement situations. When we talk about animals and introducing this technology, we have both we can have both an impact in research animals that we use or farm animals, and also in the environment when we release this kind, several type of organisms that now, as I said, can be editable. And this rationale, the rationale that is behind this is also something that is about what we call responsible research and innovation, RRI. And RRI is something that seeks to bring issues related with research and innovation into the open, anticipate their consequences, and then involve society. So we have to somehow think what the introduction of such a technology will mean, not just for the generations that we are having at the moment in our world, but also the next ones, the future generations. What kind of society will we have when we introduce such a technology like this that has so many potential, but at the same time unknowns? An RRI establishes a kind of a picture with the 6 axes, as you can see here, and then also several actors that are involved when we look upon these different axes. In our sense, of course, for the time being of the project, we couldn't address all of them. So we had to choose some of these stakeholders to address, and we identified both policymakers and research community, in this case, scientific, so researchers from the hard sciences, to be addressed. Today, I'm only going to focus on the ones that are from the research community in this sense, and leave the policymakers for another time, because those interviews will still be conducted in the near future. As I said, the way that we addressed them was by interviewing them. We use semi-structured interviews, and the only thing that we mentioned when we were about to interview them was that the topic was about genome editing and CRISPR-Cas9. No more, no less. We actually could recruit about 22 researchers, coming from different backgrounds, as you can see in the world map, many from Europe, but also some from Canada, USA, Brazil, and China. And some of them were actually working on these countries, and they were not even from there, for example. So we had some diversity there. In terms of the guide of the interviews that we used, we tried to cover some topics that are illustrated here. We tried to ask something about future of CRISPR-Cas9 technology in applications, the ranking of hypothetic applications with different organisms, and also the difference between germline and somatic genome editing, and finally the consequences of CRISPR-Cas9 animals, both on the research and farm contexts already. With that, we hope to be better elucidated on the concerns, the limitations, and the potential of such a technology like CRISPR-Cas9. And jumping to the results part, we actually identified a lot of different codes when after we conducted the interview. So we had our transcripts after we transcribed them. And then we had to identify some meaningful, let's say, parts of the text that were important for us, that were relevant. And in that sense, we divided those different meaningful parts in 10 different first-order codes to be better differentiated. But as you can see as well on the column on the right, we actually had to further code more than this, because for example, these 3 different codes are not enough. They don't explain themselves just by the name. So we actually had to code additionally and try to see what they were talking about within these different categories that we were establishing. And today, of course, I cannot talk about all of them, and also it would be very, very boring to talk about all of this, maybe for you. But what I'm going to bring for you today, and in the same line that Susana mentioned, is about ethical concerns. Also on the ethical concerns, as you can see, we identified these 7 additional codes, but I'm only going to focus on these 5, also for the time-wise consideration. So jumping right ahead to the results part. One of the things that we identified in our code, within our codes, was lack of knowledge. lack of knowledge in the sense that we identified several themes that were also differentiated here. And they are named as follows. Hard to predict the outcome on offspring, may have effects for the environment, and there are unknown impact in species. So these three themes we identified within the lack of knowledge. And what I'm going to do now is to read some of the citations, some of the excerpts of the text that we gathered to justify such theme that we identified here. So on the hard to predict outcome of on offspring, one of our interviewees, Ruben, mentions this. If you are just doing somatic gene editing or somatic gene therapy and it goes wrong in one person, you're going to have a problem in that one person. But if you are doing something that has a problem that affects the whole population, then you have a big problem. Then on the theme about the fact that it may have effects for the environment, such gene editing and CRISPR-Cas9 technology, Megan says, this gene drive to avoid insecticides used in agriculture, I guess that's also a very nice application, although I do not oversee all the ecological consequences that might happen as well. And Philip also compliments on this, saying, but the gene drive will not necessarily take over, and the trait actually will be minimized. So one thing is the technical. The next thing is that put it out will be to spread it out on the whole planet. On the last topic here, the unknown impact in species, we have this sentence from Lisa. So the importance of the biosafety, it's a concern. So it's really important to think about everything when you are working with these new species. And Vince also says something about this by saying this can be applied to animals or plants or humans. Then the off-target effects are getting crucial. This has, of course, more themes that we recognized when we were evaluating this code. One of them was to understand the function of a mutation at the genome level, anticipate the consequences for the organisms, and the unknown problems of editing transplantable organs. So lack of knowledge in this sense builds on the understand the function of the mutation at genome level, and Megan illustrates this by saying the following. Considering the cost of gaining that knowledge, we have to think about how valuable it is. It behooves us to make sure that we make our best efforts to narrow down the causative mutation and know which and be fairly confident. And Benjamin also says something about this. He says, this happened because he Jiankui, doesn't have any background in biology or biomedicine or any clinical pathology. So it was purely driven by the motivation to succeed. He Jiankui, by the way, is the maybe known investigator for you that maybe heard the news that bring to life the first gene-edited twins in China, and he has been prosecuted since that happened, and he also has been so he has been disavowed as somebody recognizable at university in the science field as well. He also had a lot of conflict of interest. So Benjamin here is talking about him. Also, on the anticipate the consequences for the organisms, Megan says, it doesn't obviate the need or reduce the responsibility in terms of applying thoughtful interpretation to the data that you get before going into animal work. And George also says something about this. He says, we have to increase the knowledge of what we do and what are the consequences that these things we are doing may have in the modified organisms. And then on the unknown problems of editing transplantable organs, Benjamin says, I mean, the inner organs, that's quite difficult to control. Especially if, like a liver, you transplant the new part of the liver that might be merged or fused with the liver that remains in your body, that might cause a lot of unforeseeable problems. Okay, this was one of the codes. The other one that we identified was purpose. And here we identified also 6 teams divided in this way. We have a theme that is about gene editing that is only acceptable for some purposes. Helping many people is something that is highly valued, and purposes matter. Also, on another side, we have if they are enough to justify purposes, how is the harm benefit of such a purpose that is given, and some purposes are just unimportant. Okay, so similar to what I did before, I'm going to read the sentences. Vince, for the gene editing is only acceptable for some purposes, says something like this. The human embryo depends perhaps on for what reason. If one can prevent a life-threatening disease, perhaps I could give it a thought. But if it's just to increase the person's skills or something like that, then I'm against it by all means. And Simon says something that complements this. He says, I think that the gene editing needs to have some more purpose, like not only to make an animal run faster, or I think these should be applied to fight diseases or to feed. On the helping many people that is highly valued, Michael says, I think this resistant crop has the impact on most people because it might reduce the use of certain chemical agents and agriculture widespread throughout the planet and have the biggest impact on the most people, whereas probably targeting of certain genetic diseases will affect just a couple of people. And Megan says something similar, but in a different way. She says, again, the number of people that I think would benefit, I think that editing to target cancer and genetic diseases would affect more people than transplanting organs. Then on the fact that purpose matters, Lisa says, I think the most important would be the intention, the goal. Of course, we need to study a lot of things, but if you have like a good intention, right? A good goal, and just go, we can try to understand these secondary concerns and try to improve. And Jane also says something about the ethical is obviously what is a disease and what is not a disease. So the ethical should be where the line should be drawn on what we should treat and what we should not treat. Reflecting on the fact if they are enough to justify these purposes, Frank says this. So you could envision that in certain very severe disease situations among both adults, but also children, you could open up for possibilities of in certain situations taking advantage of the technology as it is today. And Steve says, I think it's more interesting to the scientist to do the model, and he's talking about animal research model, because it's more related to human disease. And the races, like compared to a horse with enhanced speed and muscle, or muscle and speed, of course can have many applications, but they are not really clinical significant. On the how is the harm benefit, Frank says, if it would have negative effects on the primate, so if you would put it a little bit broader, if it's gene editing for research purposes in animals for the benefit of humans, I think that as long as the welfare of the animal is not at stake. And Steve says, again, the urge of the risk benefit. A very invasive human disease, and if you want to model that, then I think that the society would accept so minimal animal welfare issues. Whereas if it's only a cosmetic trait, for instance, people would not accept due to animal welfare. And finally, on some purposes that are important, Bruce and Selma somehow agree. They say, gene editing in horses is completely unimportant. It's a sport, it's entertainment, it doesn't have value. And Selma says, that's nothing desirable in gene edited horses to increase speed and muscles. I don't see the value in this for humankind or anything. Continuing on another subject, which is about overriding biology, we also identified 3 themes here. One, which is about gene editing might cause harm to the species. Gene editing might impair or take over evolution. And gene editing is an extension of selection. So on the causing harm to species, Steve says, in the germline editing of the animals, like just to do what you want. So this is, of course, really dangerous. So you could actually change a species. So to change or not species, so this is dangerous for the environment. A little bit confused the discourse, but nevertheless, he mentions about what is danger. And then Paul says, there will be a big effect on the species of what we can wipe out, and it can go easily out of control. So it has to be very well regulated. On the fact that it might impair or take over evolution, Irvin says, in somatic editing, where things improve the health status of the individual, whereas in the other one, the germline, is changing the evolution of species. Jay also says, in principle, I know humans can't stop evolution, but take over evolution, develop, manipulate nature as such completely. Building themselves, this is my greatest concern. In, let's say, a little bit far in the future, in 50 years or so. And on the fact that genome editing is an extension of selection, Jane says, and people have been working with animals and modifying them for centuries to produce better, right? For the kind of what people want. And so I think this is just an extension of domestication and bringing right markers to this selection. And Owen says something different in another setup. He says, what's then a difference between having milk of an edited polled cow or having milk from a cow that is naturally polled? And on the part 2 of the overriding biology, we also have the gene editing species. Gene edited species might endanger food chain and ecosystems, and the enhancement of certain characteristics is a consequence of editing. On the first one, we have Ruben saying, you are doing a modification, creating a new animal or a new animal model species that could have big modifications to that species in particular, to their progeny, and to the other species that are related to that one. And George says, if you create new species or if you remove species, you impact on the equilibrium of the whole bunch of the different interactions that we have between different species. He even adds, that's what drives evolution on one side, but we don't know whether if we change this animal, it will be completely different. On the enhancement part, Ruben says that you could try to increase fertility in a particular species because it's economically relevant. So that's one scenario. And Dean says that I don't agree with the first of in vitro genome edited embryo or transplantation of a gene edited organ body because it would disturb such as the age and also the life condition of the human being. It's totally changed in a human. The social justice topic here, we also identified 3 themes. One is about economic capacity and the imbalance of access, edited and non-edited people, and also the patent landscape and the unfair access to the CRISPR-Cas9 technology. On the economic capacity and the imbalance of access, Megan says this. I can fix my short-sightedness with glasses, but that means I have to pay a bunch of money to get glasses. Whereas maybe you could just have it fixed, that I would have to send all the money. So am I now in an economic disadvantage because I haven't? And Irvin says this. So will the top of technologies will lead to imbalances as observed with previous technology, where some countries will have access to those technology and others will not have access to these technologies? Will some wealthy people will have access to these technologies almost by default? Just because they have money and they can travel from the countries where these technologies are not available to a country where these technologies are available. And he continues on the part of the edited and not edited people. He says, if you bring germline editing, you might be created inadvertently a new class of human beings, the ones that are genetically modified and the ones that are not. And one cannot at this stage foresee if these will introduce a societal aspect. He continues, are there people that will be genetically modified? Is there a status that people will acknowledge? Is something that is supposed to be private? Or another important, and if the latter happens, will there be any discrimination in favor of people that underwent germline genetic modification? And then he also mentioned something about the patent landscape. He says, yeah, most of those patents of CRISPR-Cas, of course, here, that are held by individual scientists or academic institutions were developed using public funds. So it seems to me minimally, they should be accessible to anybody who paid taxes. And the last, I promise, the last of the subjects is about cultural differences. Here we identify themes about the ethics that influence public receptivity. We also identified the fact that religion defines perspectives and ethics. and the level of discussion around ethics and regulation are something important. And on the first one, Michael mentions that case that he saw. He says, someone asked the Chinese scientist or talked about the European conservative view there, and the guy was just laughing. Hey, there was no ethics. There's no substantial answer. He was just laughing. And Philip also says that the cultural differences among the people on this planet is rather often not recognized as a risk factor, and the challenge to create a common set of rules for how to embrace this technology. In terms of the religion, Owen says that I'm a Christian, and so how should I compare with this gene editing? Is this a bit by playing God? But then to this extent, are we doing it anyway already with our less classical ways of breeding? And he continues a little bit after. How should I say? I mean, even in cultures where there are a lot of Christian principles in it, that has formed it over all the centuries. So I guess it can get some view as well. People think about it, even if they are not religious. And the last one, the level of discussions around ethics and regulation, where Lisa says something about this. She says, there are a lot of countries involved where these people are trying to understand how the ethics is involved. And Jay says a little bit more. He says, because even if in Germany, there would be a new regulation. So what is this in Germany? And even if you did, the 28 countries of Europe in Union would never agree. That would still mean a thing because one would probably never get all countries of the world on the same consensus. Okay, so I end here about results. I will just give you the conclusions that somehow summarize all this information, despite in a very preliminary way. One of the things that I think it's actually very well mentioned by these researchers is that pursuing somatic gene editing is okay, but we should avoid germline gene editing. And here, of course, the scenario where they envision this, and because they are more familiarized with this as well, is on the health scenario situations. So everything that is about translation to the clinic and therapeutics, they are in a way justifying it in order to find it desirable. But everything that goes for the germline gene editing should be avoided. Another thing, and in another context, which is in the context of the environment, I think one of the things that is made is the fact that they elaborate on considering the effect on ecosystems and offspring. So not just by changing organisms that live in a certain ecosystem, but also what it would mean for their progeny when they survive and multiply themselves, or they reproduce, and what will this mean if they are gene-edited species? So this will have effects on the food part, and this is health related for us humans, but also among them, between animals, between organisms that cohabitate. What will this mean? Also about the food chain, as you could see previously. The third conclusion, and this one we didn't ask for it, it wasn't also predicted in our guide when we developed it, but we found out that they also mentioned something about this, about the fact that stakeholders' behavior and their own background will be crucial to see how this technology, gene editing technologies, in particular CRISPR-Cas9, will be introduced. Where will it be implemented? So particularly when they raised questions about social justice, cultural differences, even the purpose part, they are mentioning kind of criteria to be for these technologies to be used within the different applications that they find realistic or desirable somehow. So this behavior of such stakeholders will be important for the society where we are living at the moment, and also the future ones. So I leave you with all this food for thought. It's too long, I know, but hopefully there will be questions and a discussion about this. I think it's time for me to finish. So I think it's Susana now.
Thank you very much, Pedro, for this quite thought-provoking presentation. I would expect a lot of questions and comments around it. And now I will give the word to Anna Olsson. Anna, thank you very much for being with us today. Anna has a background in animal science, and she holds a PhD from the Swedish University of Agricultural Sciences. She leads the laboratory animal science group at ICBAS, Instituto de Investigação, Inovação e Saúde, and her research addresses questions of laboratory animal welfare and ethics with a focus on the impact of a new regulatory framework for the scientific use of animals on research and innovation. Thank you very much, Hannah, for being with us today, and I will give you the word. Thank you very much.
Thank you very much, Susana, and apologies for taking so long time to figure out how to switch on my microphone. It's a pleasure and privilege to be able to speak today about a subject that is, as Susana said, took place within a research project that focused on the impact of regulation on research, which was an impact project that finished a couple of years ago. And my sincere thanks to my colleagues in this project, Peter Sander, Jesper Lassen, Sandra Silva, Joana Fernandes, and Rita Neves, who have contributed to this work. And special hello to Sandra, if you are listening right now. I know you are now at ISPUP. Obviously, also thank you to the 26 researchers that are included in this interview study that I will soon come to. But I first want to give you some background on the regulation of animal research. This is something that dates back to the late 19th century. The first country to introduce a regulation of the use of animals in research was the United Kingdom, and the 1876 Cruelty to Animals Act. And the most important measures that this legislation introduced was the requirement for the use of anesthesia, a limitation on the purposes for which animal experiments could be done, and the requirement of a license for animal experiments. The resistance to having animal research regulated is about as old as the regulation is. This is Claude Bernard, who probably some of you know as the author of the first important textbook on experimental medicine. And that is what I'm citing here. Bernard writes, from the perspective of reflecting on the discussion of criticism, the critical discussion of vivisection, so the use of animals for experiments, which he considers futile or absurd, because he says, it is impossible for men, judging facts by such different ideas, ever to agree. And as it is impossible to satisfy everybody, a man of science should attend only to the opinion of men of science who understand him, and should derive rules of conduct only from his own conscience. Obviously, we are very far from that attitude today, and around 150 years have passed. But it is important to recognize that the fear of interference with academic freedom has actually had a large impact on the capacity to regulate the use of animals in research. And to take an example from a situation where there has been a thorough investigation into the background of legislation in Sweden, the fear of interference with academic freedom meant that it took until 1944 before research was included in animal protection legislation. That was at a time when there have already been decades of legislation regulating keeping of animals for other purposes than research. We still see some of this discussion on the interference with academic freedom in the context of research with human subjects, regulation of research on human subjects, academic freedom, and the Institutional Review Board. This is a report from the American Association of Universities Professors, and as you see, it's from 2013. The papers on the background photo reflect a huge discussion that has taken place in literature also over the last decade about the EU Clinical Trials Directive, about which many researchers have strong opinions on the impact that the directive is having. Now, what do contemporary European researchers think about the regulation of animal research? There is very little systematic research into this, but it is possible to form some kind of a picture from what has been written about the debate during the revision that led to Directive 2010/63/EU. So this is the now EU-wide legislation. EU has had legislation on animal experimentation since 1986. Directive 2010/63/EU replaces the directive from 1986. The discussion and the revision process that led to the directive started in earnest in 2005, and there is reporting of this discussion in these two papers published in EMBO reports. And one of those papers by Andrew Moore is in reality, it's a report of a meeting held by EMBO in 2005, where I had the privilege also of participating. So I know roughly what the atmosphere of the meeting was, and the report is also a fair representation of the discussion that took place. So the starting point and the consensus among all the participants in that discussion that represented stakeholders, not only from scientists using animals in academia and industry, but also scientists like myself working with the ethical and animal welfare aspects of animal use. There was a wide consensus that we need to work on this for a level playing field. The previous directive was very narrow, and there was a huge variation between countries who had a much more extensive regulation going much beyond the directive, to countries which had simply integrated or transposed or even translated roughly the directive into their own legislation. But there were very many concerns. It was quite a tense discussion. And the concerns were that a new directive, and this was at a point when no one has seen a text of the directive. The first draft came out in 2008. There were concerns that a new directive would put unreasonable restrictions on research, step by step to stop. This is a verbatim citation of one of the researchers who was clearly convinced that once politicians started to put restrictions on the use of animals in research, this would be just a start of stepping up the process, and at some point we would no longer be able to use animals in research. And related to that is also the concern that politicians are more sensitive to anti-vivisectionist arguments than to scientists, or related to that, scientists are not sufficiently vociferous, and this is perhaps what is the chicken and the egg here is debatable. And a view that was also expressed is, why is revised legislation necessary if scientists already follow the rules and constantly improve animal use according to the best scientific knowledge? Moving further in time, 8 years later, in 2013, when the 2010 directive was to be implemented, the fears of unreasonable restriction had disappeared. The scenario had also changed from the first draft version that the European Commission published in 2008 to the final directive in 2010. Lots of adjustments had been made to a great extent to the in response to concerns from scientists of issues that would not be practical, that would be too, that were at the moment impossible to put into practice, etc. So scientists in 2013 had much more optimistic voices, described the directive as a workable compromise between political pressure and scientific need, and a directive that does not require impossible things. In countries with already advanced legislation, there is no increase in demand. But there were also remaining concerns over bureaucracy, the time that it would take to process requests for licenses, and the possible loss of competitiveness, and also on an unrealistic focus on the replacement of animal research with non-animal methods. Now, these are all voices speculating about a largely unknown future, because we are either in 2005 when the process is starting, or in 2013 when countries are just about to implement the new legislation. And many of these voices are also expressed in a lobbying situation where there may be an interest in exaggerating a little to make one's point come across. And there was no, has not been a systematic in-depth study of what scientists think before we did the one that we ran in 2015. This was an interview study, again, semi-structured interview, so it's an in-depth rather than a broad study. We interviewed 26 bench scientists, most in academic biomedical research, but also a few from basic or animal science, so the use of or in an agricultural setting. These interviewers work in 3 EU countries: Portugal, UK, and Denmark. These selected to be countries in different places in Europe and with a different history and a different social context regarding animals and regarding the regulation of animal research. Balanced in genders, a bias towards more senior researchers, which was on purpose because we wanted people who had been in this activity for long enough to have a perspective on things. And also quite a wide range of species, even though there is a bias towards the typical laboratory mouse. The themes that were addressed in interviews and analysis are, is regulation needed? Is regulation needed? And if so, why? What does a good and well-functioning regulatory system look like? And how well does the existing regulation function? There was wide agreement that regulation is needed for 2 reasons. One is because it protects research and researchers from public criticism. And the second is because without regulation, some researchers would go beyond what is an ethically acceptable way of treating animals. Here's a Danish scientist saying it's much better for us to have this kind of system where experiments are approved by a committee with representatives of animal welfare organizations, because it takes away that kind of speculation. That must be terrible, to sit in a situation where you don't want to tell what you do, you don't want to put signs on your building, because then you know people would come and burn it or you would be attacked. And a UK scientist says, I think it protects us as well as the animals. that we have a system that's very clear. It means that we can safely say with honesty that we are doing the experiments under an umbrella of regulation that requires us to take into account animal welfare and to have a valuable purpose to the experiment. Portuguese scientist says not everyone is conscious that to get good results you need to treat animals well. A good researcher has to be conscious, but it depends much on the researcher, and there are good professionals and bad professionals everywhere. So I can't say there shouldn't be legislation relying on people's common sense. There is no common sense. That's why there are regulations. And finally, much in line with this UK scientist says, I think if we didn't have that system, then it would just be down to investigators to decide whether they want to. And, you know, there are different opinions. And I think we would see much more contrasting ways. What is good and proper regulation? In the view of our interviewees, a good regulatory system is one that is without a too long, bureaucratic and time-consuming application process, one that is run by competent people, that has reasonable and not too restrictive standards, that is has some degree of flexibility, that is consistently applied over time, that is proportional compared to regulation of other forms of animal use, and that sets a fair level compared to other countries. The view of what works often comes across through the experience of what doesn't work. So all the descriptions of a good regulatory systems are going together with concerns about deficiency in the actual system. There is a general concern about lack of flexibility, long waiting times, and unnecessary paperwork. Particularly in Portugal, there are concerns among the scientists about competent authority being slow. And in the UK, there are complaints about the lack of consistency between individual Home Office inspectors. But there are also positive views. Even though it takes time to write applications to regulatory bodies, this time may be well invested. It may lead to better research. Focus on ethics may be a win-win for research quality and animal welfare. And the licensing process is useful when it comes to defending animal experiments to the public. It's another level of bureaucracy, but I think in general it leads to better projects, says a UK scientist. I think it's fine, we spend time on it, but normally this is just making the protocols more efficient. We think we have been much better in planning and thinking before we start, says a Danish scientist. All aspects of the Home Office regulations of animal work, which includes the 3 R's, are a positive additive to science and enabling you to produce science, which first of all has justification and secondly is robust and reproducible, says a scientist from the UK. And finally, Danish scientist says, when we finally get a license to do it, well, then my speculations about if this is okay are gone. And that makes it much easier to talk with anyone about animal experiments. The negative views on the actual regulation have to do with wasting time on filling out applications and waiting for responses. A mismatch in regulation between the dynamic nature of science and the regulation system that is based on detailed license applications for 5-year projects, which may be difficult to alter after it has been approved. And also that regulation directly and indirectly imposes costs, which at the end of the day goes from the actual research or from the care of the animals. Maybe after 5 years or 4 years, there is discovered a better way to anesthetize, but then I have to use the old way because that's what we got the license to do, says one of the Danish scientists. Regulations makes my work more expensive. It is basically because things need to be checked. I mean, record keeping. We have one person working in the fish facility who is basically all day putting things in a database. That's all she does, and she has to be paid, says a UK scientist. That type of very bureaucratic regulation is not good at all. It doesn't benefit a collaboration with researchers. One should try to alert researchers. One should make an effort. one has to be careful with how one does things, and bureaucracy doesn't help, says a Portuguese scientist. And finally, a Danish scientist says, I think it's a heavy process in Denmark, difficult, awkward, and that destroys also our competition because we are way behind. I can wait a year, then everybody else is publishing, right? All my collaborators do the experiments, and I never heard them complain about a one-year process. The final topic that we also talked to scientists about is the 3 R's of animal research, which is replacement of animal experiments with non-animal methods, reduction of animal numbers, and refinement of the research with animals to reduce animal welfare problems. And I will present these 3 themes a little more superficially without going into the actual citations. Regarding replacement, the scientists that we interviewed present a number of reasons for why they actually use animals. They say they do this because when they do research into complex issues where animals give a more valid result, for situations where there are no available alternatives, they also give structural arguments for why they use animals and not non-animal methods. Some of them say their role in the whole research infrastructure and ecosystem is to do animal work, and that other scientists before them in the chain of knowledge have done the non-animal work. And they refer to pressure from journals and funders to use animals to add data from animal experiments to the body of data before something is published or before it's funded. Regarding the reduction of animal numbers, there is a widely shared concern about the risk of using too few animals, about results that need to have sufficient statistical power and therefore a sufficient number of animals. There are scientists referring to avoiding some experiments because they would require too many animals. Some argue that they reduce numbers to limit the harm that they do to animals. But others, again, refer back to the external pressure to keep animal numbers in line with what journal reviewers require in order to get their work published. And the third R, the refinement, is one which is received with a generally positive attitude. Some people point out that sometimes research considerations put constraint on refinement because there is a need to study disease conditions which are quite advanced, where the animals get really ill, but to get knowledge of that situation, one needs to bring the disease progress to that point. Some scientists see structural and economic barriers for increased levels of refinement, and some see this as a win-win through which you get better science and at the same time better animal welfare. So to summarize this study, there is a consensus on the need for regulation of animal use, and the intentions behind legislation are shared by researchers. There are critical concerns, to some degree differing across countries, so relating to a specific situation in a specific country, about how well the actual regulation functions in practice. Researchers who do experiments with animals do not consider replacement an alternative for those experiments. There is ambivalence concerning the reduction of animal numbers. There are more positive views on refinement. And there is a transversal concern about unproductive paperwork. So to bring this two talks together, what does the researcher perspective tell us? This is perhaps not the same for each of the studies, and I'll reflect on both of them separately. From the perspective of, or the background of the work, our work, talking to scientists about how they perceive regulation of animal research, we developed this thinking these are the key actors. These are the people whose activity is directly regulated. It's important to hear their view because the successful implementation of regulation depends on how it is received by key actors with a practical or strategic role in the system. And as we just saw, the attitude is favorable towards regulation if that is reasonable and functional. So to a great extent, if that regulation is protecting animals and enabling research, as is the title we chose for this review paper, published in 2016 in the ILA journal, which I recommend if you want to know more, if you're curious about how what actually Directive 2010 means in practice for European research. In the interviews with gene editing researchers, we see something about their overall attitude to the different implementations, the different applications. They see somatic and germline editing as distinct issues, and they find somatic gene editing as something that should be encouraged, germline not so much. Ethical, regulatory, and social issues of gene editing matter. They come across in these interviews. Scientists are not thinking of technical issues only. And the concerns presented by the scientists about ethical, regulatory, and social issues, they reflect largely those that ethicists and legal scholars, people engaged with the policy making and the ethical analysis of the matter, publish in literature. So the view, scientists don't have a view that is distinct and different from the rest of society, or from those who have put more thought into the philosophical and regulatory aspects of the use of gene editing. Now, what does this have to do with public health? Is also a question I would like to address, given that we are presenting this in the seminar series of the leading research institute for public health in Portugal. And I'm borrowing this umbrella view of the One Health concept as the basis for us to reflect on how what we just presented links back to an issue of public health. And I would like to invite Pedro back to comment on the gene editing research in the context of One Health.
Okay. Thank you, Anna. Yeah, I think considering what we saw with all these researches, especially on these ethical concerns topics, we saw that somehow they kind of differentiated, and looking at the umbrella, we can see that a lot of different fields are illustrated there. But I think what raises my attention are those three that are glued somehow: the individual health and the population health, along with the ecosystem one. I think those three were touched when they mentioned different issues that they have with the gene editing and with the CRISPR-Cas9 technology, especially if we look to this very well distinctive view between somatic and germline gene editing. We see that one concerns about an individual, and this is the somatic. But then if you change cells that will have an impact in generations that are still to be born, then we have a problem at the population level. So in that sense, we face some issues that are different. They are not considering the own individual solely. It also concerns the population, depending also on the environment that we are situated. I think the other part that was very well recognized by them was this ecosystem health, in the sense that we have to be careful with what we don't know and what we can cause an impact at the ecosystems level, especially if we also change different organisms. And how is that impact on their progeny, and how will they and their own descendants interact with the other species that are native, that are natural somehow. They are not gene edited. So in that sense, I think we also have here a situation where this is covered. This is also abrogated by this One Health umbrella. And I think somehow this is also depicted in the different citations that they say. The discourse sometimes when they talk about these issues are not as clear as when they talk about technical challenges. We saw that as well. I didn't present here, but technical challenges are much more their field, so they know what they are talking about. They are very clear on the speech level. When they go to these situations, of course, those are more complex questions, but they nevertheless they touched it, and as you said as well in the latest conclusions, they recognize most of the features that also are in the literature. So somehow they are connected with this. They are not just unaware and doing their technical work and not concerned at all with these different issues. And maybe they don't think about it in the sense of like this umbrella illustrates, like an interaction that is happening every day, because maybe they differentiate between the applications, but nevertheless, when they are invited to think that way, they do it. And I think that's one of the highest contributions that we had here.
Thank you. And I'd like to reflect on animal experimentation in the context of One Health. It is not something that we often talk about in the context of One Health, but really the idea that that human health and animal health are interrelated, and that they have consequences for each other, but they also have a possibility that they can contribute in a positive and a negative way towards each other. That is really the basis for doing animal experiments. And actually, if you look at this picture below the umbrella, the entire circle that's on the right side, comparative medicine and translational medicine, these are terms that we use a lot to talk about research with animals in a biomedical context. In fact, one of the main journals published in the US in the field is called, its title is Comparative Medicine, and that is all the journal. All that journal publishes is research with animals with the purpose of applications or developing knowledge for human medicine. But also a lot of the research that is done in what's in the left circle here is also research that is done on animals. It is interesting if we think about vaccines and therapeutics, which is super high on the agenda right now. And interestingly, one of the vegan organizations in the United States have issued a statement, I think for the purpose of encouraging their members to get vaccinated against COVID. They have issued a statement that's saying that even if you're a vegan, if you think no animal use is authorized, at the moment, there is no development of vaccines without animals. So I think the underlying message here is you can take this vaccine because that is the way to get vaccines to the market today. And with that, I would like to wrap up the presentation part of this. We are the Laboratory Animal Science Group at the Institute for Research and Innovation in Health, like I3S. And our research, I like to describe it as in the picture of the schematic to the right, in the interface between science, society, and animals. we have a small team that work on anesthesia and experimental procedures and the 3 R's. We have work on ethics, which is where both the talks of this presentation really fit in. And we also have work on behavior and animal welfare of laboratory animals, as well as on companion animals. And I'm going to hijack this presentation to say that we have a very important paper on dog training coming out tomorrow. So if you're interested in companion dogs, that'll be something interesting for coming out very soon. And our website is on the link at the bottom of the slide. Thank you.
Thank you.
Thank you, Anna, for this wonderful presentation. I think that we have no questions at YouTube, but I would like to invite you to a brief comment on something that is in my mind now after listening to both presentations. It seems that there is an alignment between the concerns of ethicists, law academics in the fields of law, and those researchers who are responsible for genome editing and animal experimentation in the labs. What I would like you to ask you is it seems that there is some level of agreement on normative ethics from those who already have positions as stakeholders. But the public, what we call the general public, the lay population, wouldn't they had additional information, more in a basis of a pragmatic ethics that could go behind the concerns that seem to be quite similar among those who already are in charge of conducting research, both in gene editing and animal experimentation. What do you think about, I think that you already have information about what we call civil society, and I would just wonder if there is something new and that could represent a concern for the population. And this is particularly relevant because it is very important to assure public confidence and public trust, and to have systems of accountability where all the citizens are involved. So it's more a general question about what do you think about the perspective of those who are usually not classified as stakeholders.
So that's a great question, and I'm going to pass the question on to Pedro. I will just put it in context. That this is a question that we have not addressed in our own research in the sense of collecting data, but we are just finishing a systematic review in which Pedro has done a massive job in going through the entire literature on surveys to the public that have been done by others. So against that background, I'm sure that Pedro has an answer to the question.
Yes, okay. As Anna said, very good question in the sense that we also ask the same thing. And actually it's that was supposed to be, and it is actually one of the objectives of our own project, because one of the things that we also wanted to investigate is the public acceptance. Of course, since we are conducting these interviews that are very time consuming, and since we have 2 different stakeholders to address, that part for the time being of the project would have to be left aside. But in the end, and also looking at this RRI picture that I showed, the general public is actually the latest in the sense that they would have to be also questioned on what are their thoughts about it. So the way that we try to do this, and as Anna was saying as well, is that we opted for a path to look on the different literature that exists since the first surveys have been conducted to the public on genome or genetic modification and genetic engineering. Also because the language has changed over the time. And I think one thing that we are, as Anna said, we are on the verge of ending and submitting that work. But one of the things that I think is very crucial is this distinction between what we are doing to adults and children when we want to cure diseases. And people usually have a very, somehow a very positive way of looking at things. But when this is about other health-related purposes, like using animals as a vehicle to that are genetically modified in order to have some products from them that we are going to use, both food or medical, then a different question is there. It's not the same positive view that we see as striking in the gene therapy situation. And then when it comes to the gene editing per se, so since the advent of CRISPR-Cas, some surveys have also been conducted. And we see that somehow they have a similar behavior, but they pay attention to different things when looking at treating adults, also because of this distinction between germline and somatic. They are much more aware of these differentiation. So I think this is a difference between these studies nowadays and studies that have been conducted, let's say 30 years ago, for example. And then when it comes to the gene editing of animals, we also have some differences. But you are right in the sense that we have to look on public opinion, and not just because they are the most important ones, because they will be the most affected when compared to other stakeholders, but also because all of us are those, even the stakeholders that are policymakers and are researchers are in the end part of the civil society. So we would like to know, what is it to have a technology, and can we use it to treat diseases? Which type of diseases? As some of the researchers pointed out, I don't think maybe with all of the information was remarkable, but they say, first of all, we have to distinguish what is a disease and what is not a disease. I think this line being drawn is very important. Once we establish that for the different type of diseases, because then diseases are not the same for public opinion as well, then we can maybe have something that once the technology is ready, once the technology is possibly scientifically advanced with the most of the challenges overcome, then maybe we can have a sense where we measure public opinion and we ask them more specifically than just asking in general, so what do you think about gene editing? Because maybe the first reactions will not be as positive as if we give them examples. I think we have to also pay a lot of attention to that work. Give information to the public so that the public can answer our questions. If we just ask things on something that is too broad, too general, it will be maybe hard for them to answer, and maybe they will answer more emotionally than rationally, for example. So yeah, I see that this is a matter of urgency, yes. And I agree that work should be done and continued.
I think this would also be very timely work to be done at this point, because as you know, the Nobel Prize was awarded for the discovery of the gene editing technique this year. So that's brought again renewed attention to the technology. But we are at the end of a PhD project on this, and this is not the time for us to open new research avenues. So that is out there for someone else to pick up.
Okay, thank you very much. Pedro, it seems that you already have your postdoc research, but it would be really interesting because emotions and rationality seems not to be so distinguished. And in public health, it is really important to understand how we embody this knowledge and also these challenges, and to think collectively about how to address ethical issues in these two main research topics would be really interesting. So thank you very much, Pedro. Thank you very much, Anna. It is really a pleasure to have you with us, and I wish you continuing good and excellent work as you have been done. Thank you for all those who worried with us, and you can see the webinar in the website of ISPUP in the future if you wish to, and you can also follow us at the website of the DataGov project to be aware of the most recent publications that we have also in the group. So thank you very much to all of you, and we will finish the YouTube webinar. Thank you and see you around. Okay.